Welcome to the deep dive.
Great to be here.
Today we're jumping into some really potentially game-changing stuff around illness therapy.
Yeah, we've got a mix of sources looking at things like, well, sea snail venom.
Right, and also revisiting some traditional antiparasitics. It's quite a range.
It is. The idea is to basically pull out the key information, the really interesting bits for you from all this material.
Exactly. Cut through the jargon, get to the core insights. So, where should we start? Maybe the sea snails?
Sounds good. The Conus marine snails.
Okay, so these aren't your everyday garden snails, obviously. They're predators.
Highly sophisticated ones.
And one source calls their venom. It says it has an unparalleled molecular diversity of pharmacologically active compounds. That's quite a statement.
It really is. Think about it. This venom is like a chemical weapon, but incredibly refined. It's full of these peptide toxins.
Toxins, right.
Exactly, conotoxins. And they target ion channels. Specifically, one source really focuses on nicotinic acetylcholine receptors, HERs.
Okay, NACHRs, those are ligand-gated ion channels involved in nerve signals.
Precisely. Super important for communication between nerves, and muscles. Fundamental stuff.
So the snails have evolved ways to mess with these signals.
Very precisely.
Yeah.
But here's the interesting part. The review we looked at points out that for a lot of these conotoxins, we actually don't fully know how they work.
Really? So there's still a lot of mystery there.
A huge amount. It's a frontier, really. This review tries to map out what we do know about the ones hitting those HERs. You know, the pharmacology, the structural function links.
So like how the shape of the toxin fits the receptor.
Exactly, that lock and key idea, understanding which key fits which lock.
And there's a figure 2, showing the receptor structure.
Yeah, it helps visualize it. You see the different parts, the subunits coming together, forming a pore.
And the binding sites where the toxins attach are right at the joins between these parts.
And it shows a single subunit sort of embedded in the cell membrane to get you a feel for the molecular machinery involved.
So understanding this could lead to new drugs targeting these NKRs.
That's the therapeutic potential, absolutely. If you know exactly how these toxins work, maybe you can design drugs that are super specific.
For what kinds of things?
Well, potentially things like chronic pain, maybe some neurological disorders where these receptors are involved. There are actually already some drugs derived from conotoxins used for pain.
Wow. Okay, so that's one really fascinating area. Nature's pharmacy, kind of.
Right, and that actually leads us nicely into the next topic. Parasites and antiparasitics.
Okay, shifting gears a bit.
Wow.
We looked at a source discussing ivermectin and some of the, let's say, myths around it.
Yeah, it's quite relevant, this idea of looking again at existing tools. The article tackles that myth, the horse dewormer thing.
Right, which ignores the fact that, billions of doses have been given to humans since the 80s.
Exactly. It's FDA approved for human use. It's on the WHO Essential Medicines list. It's a serious human medication.
And the Nobel Prize for its precursor, avermectin, that wasn't for, just animal health.
Absolutely not. It was a major breakthrough for human parasitic diseases.
So myth number 1, busted. What else? The idea that it can only be antiparasitic.
Yeah, the article debunks that too. It makes the point that lots of drugs have broad spectrum effects.
Like antibiotics that also have anti-inflammatory effects.
Precisely. Azithromycin is a good example.
Right.
Or even natural compounds like azadirachtin from the neem tree. They call these broad-spectrum therapeutics, or BSTs.
Okay. And this ties into drug repurposing.
Exactly. Finding new jobs for old drugs.
Uh-huh.
The source mentions nitazoxanide started as antiparasitic, found to have antiviral uses too.
So the idea that ivermectin might have other effects, like antiviral, that's not totally out of left field, pharmacologically speaking.
Not at all. And the article points out studies showing in vitro antiviral activity for ivermectin going back to 2012, way before recent events.
In vitro meaning in the lab, like in cell cultures.
Yeah, in petri dishes, not in people or animals necessarily at that stage, but it showed potential.
The source also mentioned safety, called it incredibly safe.
And pointed out it's over the counter in some places. Now, obviously, there's the whole COVID-19 controversy.
Right, and the source touched on that.
It did. It mentioned reports of positive effects, but also the media focus on allegedly fraudulent studies and claims of censorship. It presented it as a very contentious issue.
And we're just relaying what the source said, not taking a side here.
Absolutely. Just reporting the discussion points raised in the material. It even mentioned a hypothesis that maybe any effect against COVID was actually due to hitting underlying parasites.
Oh, right. But that hypothesis has been challenged.
Yeah, the article noted that too. It's complex.
But then there's another angle for ivermectin in our sources: cancer.
Yes, this was really striking. A study looking at its effect on esophageal squamous cell carcinoma, ESCC,
And it found.
potent anti-tumor activity, both in cell lines in vitro and also in mouse models in vivo.
Against esophageal cancer. That's unexpected for an antiparasitic.
Very. It seemed to inhibit the cancer cell growth that induced apoptosis.
Programmed cell death.
Right. Potentially via the mitochondria, the cell's power generators. And it also seemed to suppress the cancer cell's ability to migrate and invade tissue. Even showed anti-metastatic effects in the mice.
Did they figure out how it was doing that? The
And they
mechanism?
looked into it. They found ivermectin strongly inhibited a protein called PAK1.
PK1.
Yeah, and inhibiting PAK1 seemed crucial for the anti-cancer effects. Ivermectin apparently promotes the breakdown of this PAK1 protein.
Okay, that's getting quite specific.
It is. And one more thing from that study, it seemed to work with standard chemo drugs.
Like cisplatin and 5-fluorouracil.
It showed a synergistic effect, meaning the combination was better at killing the cancer cells than either drug alone.
So, again, it comes back to this drug repurposing idea. An old drug, new tricks.
Exactly. It really makes you think about what else might be sitting on the shelf.
Definitely. And thinking about existing things, our sources also briefly touched on more traditional or natural approaches like black walnut.
Right, connecting back to that anti-parasitic theme, but from a natural product angle. We looked at a study on walnut constituents.
Not just antiparasitic, though, right? It mentioned other benefits.
Yeah, antioxidants, anti-inflammatory properties,
Mm-hmm.
and potential roles in personalized cancer prevention and even the gut microbiome.
The microbiome seems to pop up everywhere these days.
It does. This study found walnut supplementation helped protect against colon damage in mice, possibly through metabolic changes.
And another mention, protection against stomach ulcers and benefits in an H. pylori cancer model.
Yeah, anti-inflammatory and anti-tumorigenic effects in that context, too. So walnuts seem to have quite a range of potential benefits being studied.
Okay, so we've covered a lot of ground here, from highly specific toxins in sea snail venom
Targeting those nicotinic receptors.
to looking at an established drug like ivermectin in completely new ways, for cancer even.
And touching on natural compounds like walnuts, too.
It really shows that illness and therapy are complex, and answers might come from, well, unexpected places.
Absolutely, from deep-sea venom to the pharmacy shelves to the health food store, potentially.
But it's also clear that there's still a lot to learn, right? The exact mechanisms for the conotoxins, the full picture on ivermectin's effects.
Definitely. These sources raise possibilities, but also highlight the need for more research. Lots of questions remain.
So maybe a final thought for you listening. Considering all this, the history of natural compounds, marine pharmacology, drug repurposing, what other surprising places might hold keys to future treatments?
Yeah, where else should we be looking? It makes you think about how connected different fields are, doesn't it? From marine biology to oncology. Keep an open mind, I suppose.